Good afternoon, learners. Welcome you all to the today's discussion. Now, today I have taken a topic on restriction endonucleases and gene cloning. Now, this particular topic, this you will find in your BSc Botany program. The course code is BBYET 143. Economic Botany and Plant Biotechnology, and you will find this topic under Unit 14, that is Recombinant DNA Technology. So I will be talking on a part of this unit because it's a very vast unit, and I will talk on one aspect, that is restriction endonucleases and gene cloning. Now, to begin with, I should first explain what is meant by gene cloning. What is meant by clone in molecular biology or biotechnology? Clone means when you insert a particular foreign DNA fragment into a particular vector sequence. That means you are introducing a foreign DNA. It can be a gene, it can be a piece of DNA, or it can be any foreign gene into something which we call as a vector. And in molecular biology, in most of the cases, the plasmids are normally used as vectors. And I will be talking on the characteristics of the vectors and the characteristics of the plasmid. So you insert this particular foreign DNA or a gene into a vector, and then you insert it into a particular host, which in most of the cases is a bacterial cell, that is Escherichia coli, and then you have to select the clone. That means based on some markers, it can be antibiotic marker or it can be any other marker gene which will generate a color, or because of the insertion there may be a loss in color production, and then you select those recombinant clones. We call it recombinant DNA. That means whenever there is any insertion of foreign DNA in a vector, we call it a recombinant vector. And then you select those recombinant vectors, or you select those clones, the proper clones, and then you can maintain them, you can multiply them, or you can maintain them. So essentially, gene cloning can be defined as the isolation and amplification of an individual gene sequence by insertion of that sequence into a bacterium. First you insert it in a vector, and then you introduce the vector into a bacterial cell, that is Escherichia coli or E. coli, and then you grow the E. coli cells based on some selection pressure. It can be antibiotic, and then you choose the proper clone and then you multiply it. You maintain the clone and you multiply it for your future works. So the basic events are isolation of gene of interest. I will be talking on each of these steps in details. Isolation of the gene of interest. DNA fragment to be cloned is incorporated into a small replicating, usually circular DNA molecule, which we call as a plasmid vector. And the vector with an incorporated gene is called recombinant vector. That means whenever the gene of interest is introduced into that vector, you call it a recombinant vector. Then the third step is the recombinant vector is introduced into a host cell by transformation. I will talk on this technique also. Cells that have acquired recombinant DNA molecule are selected, and the recombinant DNA molecule is then multiplied. So each of these steps I will be elaborating, but before going into the detailed steps, we should first try to understand what are the components that are required for carrying out a gene cloning process. So you need to cut the vector. You need, so that because you want to introduce a foreign gene into a circular plasmid or a circular vector molecule, you need to cut the vector molecule or the plasmid molecule, and for that you need some enzymes. And the enzymes which we use in these cases are called restriction endonucleases. So you have to cut the vector, and you have to also cut the foreign gene which you want to insert with the same enzyme, that is restriction endonuclease, and then you introduce it, you join it, and for that joining purpose, you need another enzyme called ligase. Okay, so two enzymes are part and parcel of this cloning procedure, and therefore, before going into the cloning procedure, I will first take up these two components. First is the restriction endonucleases. Now, why these enzymes are called as restriction endonucleases? Endonucleases, endo means which cut within. They do not cut at the 5' end or the 3' end of a DNA. They cut in between the DNA. That is endo. Nuclease means which will cut the nucleic acid, that is DNA. So endonuclease term is very clear. Restriction, why the term restriction? Restriction means they will cut only at some specific sequences. There are several restriction endonucleases. There are several such enzymes, and most of these enzymes are actually isolated from bacteria. Okay, and each of these enzymes will cut the plasmid DNA molecule at some restricted sites or specific sites, and therefore they are known as restriction endonucleases. So these enzymes make site-specific cuts in the DNA. It's not random. Usually the enzymes that we use for cloning, they do not cut at random. They cut at some specific sites. The first enzyme was isolated from Haemophilus influenzae in 1970. According to the recommendation of Smith and Nathans, each enzyme is represented by a 3-letter code in italics. Now, how do you name? Supposing you have identified or discovered a particular enzyme, how will you name that enzyme? The name will be based on the bacteria from which you have actually isolated the enzyme. Like one example, Haemophilus aegypticus. The enzyme that you isolate from this bacteria will be named as HAE. That is from Haemophilus HA, okay, from Haemophilus H and from Aegypticus AE. So you have to understand that when you write HAE, you have to mention it or write it in italics, because it's a scientific name. The name of the bacteria, it is actually a scientific name, and therefore the enzyme name is based on that, and therefore it has to be italicized. This is mandatory. So it's a 3-letter code. Any enzyme name, restriction enzyme name, is a 3-letter code based on the genus and the species name of that particular bacteria from where you have isolated the enzyme. Sometimes a 4-letter code is used when the enzymes are isolated from different serotypes, like HIN-F. HIN, the enzyme is isolated from Haemophilus influenzae serotype F. Therefore, you are using small f, but you are not italicizing F. Mind that F is a normal one. And sometimes you often use Roman numerals like 1, 2, 3, etc. For example, EcoR1. This is a very common enzyme. Eco, that is Escherichia coli. It is isolated from Escherichia coli. Serotype is R. And what is 1? 1 means the first serotype. If it was isolated from second serotype, you would have named it as EcoR2 and so on. The next very pertinent question which is often asked is, now as I said that these enzymes, they cut DNA. They make site-specific cuts. I'm coming to what is meant by site-specific cuts. But the question is, these enzymes are isolated from bacteria, as I have said. Then they must be present in the bacteria. Then the question is why these enzymes do not cut the bacterial DNA. Okay, the bacteria is having the enzymes. The bacteria is having its own, the plasmid DNA is also there, or it has its own chromosomal DNA as well. Then why these enzymes are not cutting? The cutting or the digestion is being restricted through certain modification of bases. We call it restriction modification. So the bacteria, how does it protect its own DNA? It protects its own DNA by doing certain modification, base modifications. the restrictive host must protect its own DNA from degradation and its DNA must be modified. Usually this kind of modification is undergone through methylation process because certain bases are being methylated and therefore the bases are being protected from digestion or restriction enzyme digestion. So this is called as restriction modification or host modification. Now these restriction endonucleases can be mainly categorized into three types: type 1, type 2 and type 3. Now in gene cloning process, in biotechnology or molecular biology, we normally use type 2 restriction endonucleases, but at the same time there are type 1 and type 3 also, which you need to understand and you need to compare with the type 2 restriction endonucleases. So what are the characteristics of each of these types of restriction endonucleases? Number one is the type 1. Type 1, they are complex nucleases, example ECO-K and ECO-B. They function simultaneously as an endonuclease and a methylase. So they have both the function, endonuclease function as well as the methylase function. And they require ATP, magnesium and S-adenosyl methionine as the cofactors. So I hope all of you understand what is meant by cofactor. Because sometimes the enzymes, they need certain additional components so as to function more efficiently. Okay, we call them as cofactor. Usually sometimes these cofactors are certain ions. Okay, it can be cations. For instance, magnesium, it is a very important cofactor which is often used in different enzymes. Even in the respiratory enzymes, you find the involvement of magnesium as a cofactor. S-adenosyl methionine, in short we call it SAM. ATP is a very important component which is often used. They are single multifunctional enzymes with three different subunits: restrictive subunit, modification subunit and a specificity subunit, which determines the recognition specificity. The recognition site is usually a 15 base pair sequence which can be methylated at the adenine position in the recognition site, and the cleavage site is approximately 1000 base pair away from the 5' end of the sequence. That means the methylated site and the recognition site, the cleavage site and the methylated site, they are not same. Okay, they are distinct. The cleavage site is located, as you have seen, 1000 base pair away. DNA molecules with recognition sequences methylated at both adenine residues on either strand are resistant to type 1 endonucleases. Cleavage occurs when the recognition site is unmethylated. So only when the cleavage site, only when the base is unmethylated, then only there will be a cleavage. So the cleavage will not occur or digestion will not occur at the methylated site. So these two sites are very much distinct. These enzymes exhibit high ATPase activity. For each phosphodiester cleavage, more than 10,000 molecules of ATP are hydrolyzed. These enzymes show specificity for the recognition site but not for their cleavage site. This produces a heterogeneous population of DNA fragments. In view of this, it is very undesirable to use in the gene cloning technique. Why we don't use type 1 restriction endonucleases? Because they normally produce a very heterogeneous group of fragment or heterogeneous population of DNA fragments. And that is why they are very much less used in molecular biology. Next, we move to type 2 endonucleases. Now, type 2 endonucleases, you have to pay special attention to the type 2 endonucleases because these are mostly used in molecular biology. Now, they are simple enzymes that consist of a single polypeptide. They have separate endonuclease and methylase activities. So, in this aspect, they are same like type 1. They recognize a specific nucleotide sequence and cut a DNA molecule at this sequence and nowhere else. So, for each enzyme, there is a specific DNA sequence. It is not random. It is very specific where it will cut. Like EcoR1. If you take EcoR1, it will have a specific sequence. If you take another enzyme, BAMH1, it will have another sequence. If you take another enzyme, HINDI3, these are all names of different restriction endonucleases. It will have a different site. So, usually most of these restriction endonucleases have distinct recognition site, and they will cut only at that site and not elsewhere, ideally. And these sequences can be tetranucleotide, pentanucleotide, hexanucleotide or octanucleotide sequences. Mostly these are hexanucleotide sequences. That is, hexanucleotide means that is which is a sequence which is composed of 6 nucleotides or 6 bases. PVU1 isolated from Proteus vulgaris cuts DNA at a hexanucleotide CG80CG. PVU2 isolated from the same bacterium cuts at a different hexanucleotide. So PVU1, PVU2, they differ in the cleavage site. They are very stable and they require magnesium as cofactors. Majority of the sequences, now this fourth point is very important. We call them a palindromic sequence. What is palindromic sequence? That is sequences which read the same in both the direction. If you read this sequence from 5' to 3' end, and if you read this sequence from the reverse strand, if you see the picture, or if you see the sequence that I have mentioned, that is EcoR1 sequence, see GAA-TTC. If you read this sequence in the reverse order, it will be the same, GAA-TTC, based on the complementary base pairing. So such type of sequences are called as the palindromic sequences, that is, they have an axis of rotational symmetry. And most of the type 2 restriction endonucleases, they cut at palindromic sequences. That is, they have an axis of rotational symmetry. Many restriction endonucleases make a simple double-stranded cut in the middle of the recognition sequence. Sometimes it happens that they do not have an axis of rotational symmetry. They cut blindly, or they cut at the middle of a recognition sequence, resulting in a blunt or flush end. We call such enzymes as blunt cutters, like PVU2, ALU1, HAE3, even SMA1, SMA1. So these are blunt cutters. Other restriction endonucleases which do not cut the two DNA at exactly the same position, but they make staggered cuts. Staggered cut means the cleavage is staggered usually by 2 or 4 nucleotides, so that the resulting DNA fragments have short single-stranded overhangs at each end. So they will cut in such a manner that there will be an overhang at the two ends. Mostly 5' end of the complementary strand, there will be an overhang. That is, it does not cut in between. It cuts in such a fashion, for instance, a hexanucleotide sequence, it will cut in such a fashion that there will be an overhang at the two ends. The staggered cut may result in overhanging ends either at 5' like EcoR1 or 3', that is PST1. The 3' end always carries a hydroxyl group, whereas the 5' end always carries a phosphate group. That we all know, that the DNA always has a 5' phosphate group and a 3' hydroxyl group. Some restriction endonucleases possess identical recognition and cleavage sites, and such enzymes are called as isoschizomers, like Hindi3 and AHSU1. These two enzymes, they are called the isoschizomers because they cut at the same sequence. Some enzymes possess the same recognition site, but they differ in the cleavage site. Like SMA1 and XMA1, they have recognition site same, that is CCC-GGG, but their cleavage sites are different. Where exactly they will cut, that is different, although the sequence is similar. Ninth point, some restriction endonucleases with different recognition sequence may produce the same sticky end. This is also very interesting, like BAMH1. It has the sequence GG80CC. BEGL2, BGL2 or BEGL2, it has the recognition sequence AG80CT. And SAU3A, this is another restriction enzyme. Its recognition sequence is G80C. However, all these enzymes will cut their respective sequences in such a fashion that they will generate the same sticky end, that is GATC. So we call them to be compatible enzymes. If you see this picture now, so what is meant by a staggered cut? As you can see that there is an overhang over here. The cleavage occurs in such a fashion that if there is a cut here and if there is a cut here, then automatically there will be an overhang. And blunt end, it will cut exactly in between so that there is no overhang. And the table that I have given, it's a table which is showing the names of different restriction enzymes like the staggered cut enzymes, the blunt cut enzymes, and for each of these enzymes, you have the producing organism or the source organism, which is usually a bacteria like BAMH1. It is isolated from Bacillus amyloliquefaciens, serotype H. Then EcoR1, as I already said, Hindi3, Haemophilus influenzae. So in that way, there are the nomenclature of the restriction enzyme is given, and here you can see the different recognition sequences. As I said, for each enzyme, usually there is a distinct recognition sequence and a distinct cleavage site within that recognition sequence. Lastly, the number of type 2 enzyme cleavage site, it depends on the size of DNA, its base composition, and the GC content of the recognition sequences. So it depends on a number of factors, the number of the cleavage sites. Another very important point with regard to restriction enzyme digestion is the star activity. Now, before discussing star activity, let me mention that why this star activity actually originates. Now, whenever you are setting up like in laboratory, if you want to set up a reaction, so what you have to do, you have to take the plasmid DNA, you have to take a microcentrifuge tube or a small tube where you will set up the enzymatic reaction. That means you have to add water first, of course, and then the plasmid molecule or the DNA molecule. Then you have to add the buffer which is specific for that restriction endonuclease, because you all know that any enzyme, it will need a buffer. So you have to add the buffer and then you have to add the enzyme. So all of these have to be added in proper proportions. I am not going into that part because that is the more sort of technical part or the practical part. But theoretically, you have to add all these components and then undergo the enzymatic reaction. How do you undergo an enzymatic reaction? You have to carry out the reaction at 37 degrees centigrade. That means you have to take a water bath. You have to place the reaction mixture in that water bath at 37 degrees centigrade for, let's say, 2.5 to 3 hours or maximum 4 hours, not more than that. What will happen if you keep it for prolonged duration? Then what will happen? The same enzyme, for instance EcoR1, it cuts at a specific site, but if you prolong the duration, then it will start making unspecific cuts. That is, it will start making random cuts. It will cut the DNA at those sites where it was not supposed to cut. Or if you change the buffer composition, or if you don't maintain the reaction conditions properly, temperature, buffer, and all other prerequisite conditions, then what will happen? The enzyme will make non-specific cuts. And that non-specific cut, we call it as a star activity. So as you can see here, that star activity, it depends on the reaction condition. If you change the pH, if you lower the concentration of NaCl, replace magnesium by manganese, and also due to methylation of DNA, all these are factors which might lead to erroneous restriction digestion. Like EcoR1, normally it recognizes GAA-TTC, but if the reaction condition change, it recognizes a tetranucleotide sequence AATT. So it will make erroneous cut or wrong cuts in the DNA molecule, and all your work will actually fail. Okay? So you have to make sure that you maintain the reaction condition, or anyone who is doing the experiment should maintain the proper condition. Next, I quickly go to type 3 endonuclease. Why quickly? Because this is also not very much used in gene cloning purposes. So they have two different subunits. One unit is responsible for site recognition and modification, while the other is responsible for nuclease action. They also require ATP and magnesium as cofactors. Recognition sites are asymmetric. These nucleases cleave DNA at specific sequences which are non-palindromic. So this is the difference with the type 2 restriction endonuclease. Cleavage takes place by nicking one strand in the immediate vicinity of the recognition site, usually 25 to 27 base pair downstream of the recognition site. In contrast to the staggered ends, the single strand ends produced by type 3 always differ from each other and thus cannot be recombined at random. So here you don't have the staggered ends, you have the single stranded ends. This is another difference. They lack ATPase activity and S-adenosyl methionine is not required. This is also another difference. This is a difference with the type 1 where you need these components. They produce a homogeneous population of DNA fragments, unlike fragments obtained by cleavage of type 1. In type 1, we had a heterogeneous population of fragments. Here we have a homogeneous population of fragments. They are not used for gene cloning because cleaved products are not uniform. So the ultimate, the most important component of restriction endonuclease or most important type of restriction endonuclease is therefore type 2. And mostly we use different types of type 2. Now, which enzyme you will choose for your work, that will depend on which enzyme sites are present in your vector molecules. I will come to that. I will show you some of the pictures of vectors or a plasmid DNA molecule which will help you to understand regarding the choice of the restriction endonuclease. Before that, we come to the next component of cloning, that is another enzyme which I already mentioned we need, that is the DNA ligase. That is, once you cut the DNA, you need also to join the DNA. That means the plasmid vector and the gene of interest, these two need to be joined. And for that, you need one enzyme called the ligase. Now, the enzymes used to join DNA fragments, and how this joining is accomplished by ensuring the formation of the phosphodiester bond. Phosphodiester bond between the 5' phosphate and the 3' hydroxyl group. When the vector and the foreign DNA are both cut with the same restriction enzyme, the overlapping ends of the vector and the foreign DNA are compatible and complementary. So these DNA fragments, so now you are just mixing the two. You are mixing the foreign DNA and you are mixing the foreign DNA with the vector DNA. Both are cut. And therefore, there is sticky end generated. And when you add ligase in presence of again another buffer, which is ligase buffer and ATP, then there is a joining. Okay, and that joining is known as the ligation. And the enzyme which you use for this purpose is called ligase. So ligases act on DNA substrates with 5' terminal phosphate groups and form the phosphodiester bond between the two DNA sequences. This is the final step in construction of a recombinant DNA. So for recombinant DNA production, you need to add the ligase enzyme, and this process is called ligation. Now, ligase can be of two kinds: E. coli DNA ligase and bacteriophage T4 DNA ligase. Both these ligases, they encode ligases which can seal the single-stranded ends, or we call it nicks. Nicks means cutting. Single-stranded nicks between adjacent nucleotides. The reaction catalyzed by the enzymes of E. coli and T4 is very similar, but they differ in their cofactor requirements. The T4 enzyme, it requires ATP. E. coli enzyme, it requires NAD+. So basic mechanism of action is same for both, but they differ in their cofactor requirement. Both of them, they catalyze the same reaction, that is, they catalyze the formation of phosphodiester bond between the 5' phosphate and the 3' hydroxyl group. In each case, the cofactor is split and forms an enzyme AMP complex. Complex binds to the nick and makes a covalent bond in the phosphodiester chain. But which is more common in use, whether E. coli DNA ligase or T4 DNA ligase? Answer is T4 DNA ligase. Why? Because T4 DNA ligase can join DNA molecules of anyl cohesive termini or nicks, as well as join blunt-ended double-stranded DNA molecule. You remember I just talked about the two different types of cuts. One is staggered cut, another is blunt cut. So the T4 ligase is capable of joining both quite efficiently, almost in an equally efficient manner, which is not the case with E. coli DNA ligase. So T4 DNA ligase is more preferred for blunt end cloning as well as for staggered cut cloning or overhangs, those cuts which are having the overhangs. Another enzyme which is often used is known as the alkaline phosphatase. Now, alkaline phosphatase, what is meant by phosphatase? That means an enzyme which will remove the phosphate group. Okay. Now, where is this phosphate group located in DNA? It is located at the 5' end. So, from the 5' end, you need to remove the phosphate group. Now, you may ask a question, why? Why we need to remove the phosphate group? Because you don't need self-ligation. Like, if you cut one vector molecule with EcoR1, right? Or if you cut any DNA with EcoR1, and if there are, you need to clone something for any cloning purposes. If you are cutting it with a single enzyme, right? Let's say EcoR1 or let's say Hindi3, then it might happen that a certain percentage of the vector molecule is self-annealed because it is having the sticky ends. So the 5' end and the 3' end, okay? So there can be a recircularization of the plasmid DNA due to self-annealing, and you don't want that self-annealing to happen. And therefore what you do, before the cloning procedure, you need to remove the phosphate group from the 5' end of the DNA. And that is accomplished through another enzyme called alkaline phosphatase treatment, which will remove the 5' phosphate group from the DNA. But mind that you need not always use this enzyme for cloning, because only when there is a chance of recircularization of DNA, okay, then only you use this particular enzyme. If you are cutting the vector molecule with two different enzymes, then there is lesser chance of self-ligation. And in that case, you need not treat it with alkaline phosphatase. But if you are cutting it with one single enzyme, there can be a higher chance of recircularization. And therefore, there you might need this treatment. So by treatment with alkaline phosphatase, both recircularization and plasmid dimer formation are prevented because ligase cannot join the ends. Only when you apply the foreign DNA, then only there will be, and you want that actually. You don't want the self-ligated DNA. You want only the recombinant DNA, which will have your gene of interest in the vector. And therefore, to increase the probability of getting that, you are doing this kind of treatment. So if you take a look at this figure, so here I have placed side by side the mechanism for the DNA ligase and the mechanism for alkaline phosphatase. So DNA ligase, here an enzyme AMP complex is formed which binds to a nick bearing 3' hydroxyl and 5' phosphate groups. The AMP reacts with the phosphate group. Attacked by the 3' hydroxyl group on this moiety, it generates a new phosphodiester bond. And so this is actually the mechanism for the ligation. And here what we are showing is that you treat this with the alkaline phosphatase. So what happens is this phosphate group, see the phosphate group is removed. Okay, so if you now add the ligase, then there is no chance of self-ligation. Okay, but here if the phosphate group is there and you are applying the ligase, then there is a chance that the vector molecules can recircularize and it can form a dimer. And therefore we normally go for alkaline phosphatase treatment during single cut digests, when you cut the vector with a single restriction endonuclease, to avoid this recircularization. Now with that part is over, when that part is over, we come to the next part, another component, that is vector molecule. So we have been talking about the enzyme part first. That is the restriction endonuclease, the ligase and the alkaline phosphatase. And now we come to the next part, that is the vector. That is the gene of interest you need to clone. Where you need to clone? It's a vector molecule, and usually the vector should have some properties. Number one, it must contain a replicon, or it must contain an origin of replication. Okay, each vector molecule should have an origin for replication, and therefore it will replicate in the host cell. Number two, it should have a selectable marker gene. In most of the cases, these are antibiotic resistant genes. Why? Because you need to grow the recombinant vector. After transforming it into a bacterial cell or E. coli cell, you need to grow the E. coli cell in presence of antibiotic in order to understand which are the positive clones. And for that, there should be some antibiotic resistance gene in the vector which will confer the antibiotic resistant property to the transformed bacterial cell. So ampicillin resistance and tetracycline resistance. It should have a unique cleavage site within one of the marker genes, so that insertion of foreign DNA, sometimes what happens is we do the selection in a different way. For instance, there can be a marker gene. I will come to this. We have a marker gene called beta-galactosidase. So if you insert any foreign gene within that marker gene, then what will happen? That marker gene will become inactivated. And therefore, the marker gene will not be able to exert its normal activity. And based on that, you can select the clones. Okay? And this type of selection is mostly based on some colorimetric or some color-based assay system, which I will come shortly through. This is a process which we call as the alpha complementation in molecular biology. For the expression of cloned DNA, the vector DNA should contain suitable control elements such as promoters, terminators, and ribosome binding site. This is more common with the expression vectors, not the cloning vectors, because in expression vector you need to express the protein. And therefore you need a certain promoter, you need a terminator sequence, you need a ribosome binding site, you need a start codon and stop codon. Okay, so all these components should be there. Okay, so these are the properties of vectors. And then the most important kind of vector, although in molecular biology there are different groups of vectors. Maybe in some other day or some other presentation, I will talk on the different types of vectors. But today, I will take the most common vector, that is the plasmid vector. So perhaps we all know, even at your +2 level, you must have studied about the plasmids. So these are double-stranded closed circular DNA molecules which exist in the cell as extra chromosomal units. That is, they are not component of the normal DNA or normal chromosome. They are present beyond the chromosome, extra nuclear or extra chromosomal. Although in bacteria we don't have a true nucleus, but we have a genophore, and this is beyond that genophore. This is beyond that nucleoid. They are self-replicating and found in a variety of bacterial species where they behave as accessory units, and mostly they are circular DNA molecule, double-stranded circular DNA molecule which can replicate autonomously. That means their replication is not dependent on the nuclear machinery, okay, or the chromosomal DNA machinery. It is independent of that. They are inherited independently. They can range in size from 1 to 200 kilobase pair, and most of the plasmids that we use for cloning purposes, they should not be of very high molecular weight, because the rate of transformation, it actually diminishes if you go on increasing the plasmid size. Therefore, the plasmid size, try to keep it restricted. Usually we prefer small sized plasmids for cloning purposes. Now, these plasmids can be single copy plasmids that are maintained as one plasmid DNA per cell, or multi-copy plasmid that are maintained as 10 to 20 genomes per cell. So, of course, we will prefer the multi-copy plasmid. And how this copy number of the plasmids are determined? By the rate of replication. If the replication process is very much relaxed, we call it a relaxed plasmid. That means those plasmids will have a very high copy number. And if the replication machinery is very, if the copy number is less, we call it a stringent plasmid. So accordingly, you can categorize the plasmids into two groups based on the nature of replication process, that is relaxed replication or relaxed plasmid and stringent plasmid. Now, the properties of a plasmid vector. So, in order to undergo the cloning process, number one, reduction in size of vector to a minimum. As I told you, don't go for plasmids which are very much large in size. Usually larger than 15 kilobase pair, it is very difficult to manage. So usually keep it restricted. And mostly in the cloning vectors that we use, they are mostly 3 to 4 kilobase pair range. In any case, you can go up to 10 to 12 kilobase pair, but try not to exceed beyond that limit. It should contain an origin of replication. Again, I said origin of replication should be strong enough so that the plasmid can replicate better. And therefore, you need to choose mostly the multi-copy plasmid or high copy plasmid. Third, introduction of selectable markers. It should have some marker gene, mostly the antibiotic resistant marker gene. There should be a multiple cloning site or polylinker site. That means that site should have clusters of several enzymes so that you can cut the plasmid DNA at those sites and then you can clone a desirable gene. So we call it a multiple cloning site. That means where you can clone a series of foreign DNA fragments into the plasmid vector. And obviously it's a cluster of different restriction enzyme sites. Fifth point is incorporation of auxiliary sequences such as visual identification of recombinant clones by histochemical tests. As I told you, based on some colorimetric kind of assay system, which I am coming right now. Next is alpha complementation. Now, what is this colorimetric assay? Try to understand it very precisely. It's a very interesting kind of thing. One way of selection of recombinant clone, as I told you, is you grow the recombinant clones, recombinant bacterial clones in an antibiotic medium. The non-transformed cells or the cells which have not taken up the plasmid DNA will fail to grow, okay? The wild type cells will fail to grow in the antibiotic, so they will die. Only the cells which have taken up the plasmid DNA via the transformation process will be able to grow on the antibiotic medium. So you can select. Another way of doing this is, so this is antibiotic based screening. Another way of screening is, as I told you, you have any marker gene in the plasmid backbone and then the cloning is carried out in such a fashion, the insertion of the DNA, foreign DNA into the vector is carried out in such a fashion that you clone the foreign gene within that marker gene. And let's say that marker gene, it encodes one particular enzyme which is capable of degrading one substrate which can generate a color. Now, if you clone your gene of interest within that marker gene, the marker gene becomes inactivated. The enzyme becomes inactivated. Therefore, the enzyme will not be able to degrade the same compound which you add in the media. And therefore, if there is no degradation, there is no color. So, the clones or the colonies which you get are white in color, in contrast to the other clones which are colored. So, based on the color production or color absence, you can determine which are the positive clones and which are the negative clones or which are the wild type cells. So, this is a very interesting technique and this is called as alpha complementation. However, not all vectors will have this marker gene, but few vectors, they carry this kind of mechanism. And therefore, it becomes very easy for you to clone a particular gene because you can select very easily which are the positive clones. So, the beta-galactosidase of the lac operon has been used as a tool for the identification of clone. Beta-galactosidase can be measured. How do you measure beta-galactosidase? If you add in the media 5-bromo-4-chloro-3-indolyl beta-galactoside, that is XGAL, in presence of one inducer, that is isopropyl thiogalactoside IPTG, which is a non-metabolizable allolactose analog. So what will happen? if the beta galactosidase is functional, then it will be able to degrade this X-gal, and therefore it will generate a blue color. Okay, so you will have a blue colony on agar plates. But if the enzyme becomes inactivated, why? Because you have cloned a gene of interest within beta galactosidase. Therefore, beta galactosidase fails to function. It is inactivated. So it is not able to degrade the X-gal, and therefore there is no color. So the white colonies will indicate positive clones, and the blue colonies will indicate negative clones. That means the gene of interest has been cloned within the beta galactosidase coding sequence, and therefore it is inactivated. Now, we take one representative vector backbone like PUC. Now, P stands for plasmid, UC stands for University of California. So this vector was actually designed, or it was developed by the University of California by Messings and his colleagues, and therefore the vector name was given after the name of the university, that is University of California, PUC. Now you can see that this is again a circular molecule. It is 2.7 kilobase pair with called E1 origin of replication. It has ampicillin resistance gene and a new gene called LAC-Z or LAC-Z, and that LAC-Z is the same as the LAC operon, which actually codes for beta galactosidase, and therefore there is the alpha complementation screening process available for PUC. The recombinant PUC can be created by inserting a new DNA into the LAC-Z or the LAC-Z. Cells harboring normal PUC are ampicillin resistant and are not able to synthesize beta galactosidase. When IPTG and X-gal substrate are added, it will be broken by the enzyme, and therefore a deep blue color will be obtained, deep blue colonies will be obtained. But the recombinant PUC plasmids, because you have cloned the gene within the LAC-Z gene, so it will not be able to encode the functional beta galactosidase, and therefore there will be no degradation of the product, and therefore the colonies obtained will be white. So PUC is a very widely used cloning vector because it has this facility. Of course, you can also select the recombinant clones based on the antibiotic, based on the addition of the antibiotic like ampicillin or whatever, mostly ampicillin in this case, in the media. So I hope I could make you the basic components. Which are the components that are required for cloning? I hope I could make you understand. Now I am going to the last part. Maybe in another 7 to 8 minutes, we will be discussing the steps again in a better way, because now you understand all the components and before we end up this discussion. Now, so what are the steps of the cloning process? First one is the isolation of the gene of interest. What is the gene that you are going to clone? How will you isolate the gene? So you need any source. It can be a plant source. It can be, because this is a module this is a botany program. So I will take one plant, representative plant specimen. So you can take any plant. You can take the genome. Either you can take the genomic DNA or you can take the RNA. So if you take the genomic DNA, then you need to clone a particular gene. So you need to know the sequence of that particular gene. Like let's say I want to clone a particular gene which encodes the catalase enzyme. So what I have to do, I have to know the sequence for the catalase, and then I have to actually clone, or I have to amplify through polymerase chain reaction. I have to amplify the catalase gene sequence from the genome based on PCR using the site-specific markers, site-specific oligonucleotide primers, or you can amplify the gene from the RNA molecule. That is, you isolate the RNA, you convert the RNA into cDNA through reverse transcriptase, and then you can undergo polymerase chain reaction using the site-specific primers, okay, the 5' end and the 3' end primer, and you amplify the gene. So in that way you get the gene. Mostly you use the PCR technique. Next, you need to clone the gene. But before that, what you have to do? Before that, you have to cut the gene. Either you can make a single cut, as I mentioned. You can use one enzyme, or you can use double enzyme. Okay. So when you amplify the gene, okay, through PCR, you have to include those restriction sites. So you have to design the oligonucleotide primer in such a fashion that you include those restriction sites, the 5' end and the 3' end, so that those are automatically being generated in the amplified sequence. But make sure that those enzyme sites which you are using for cloning are not present in between the gene, because otherwise what will happen? When you are cutting the gene of interest, then there will be a cut in between also, and therefore you will not get the full length gene. So you have to ensure that those enzymes which you are using are not present in between, right? Next, you choose a plasmid vector, and you have to cut the plasmid vector with the same enzyme. If you have cut the gene of interest with, let's say, HindI3 and EcoR1, then make sure you have to cut the plasmid vector also with HindI3 and EcoR1. And of course, before that you have to check whether the plasmid multiple cloning site really has those two enzymes. So there are several things which you need to check before you go for the selection. Selection of the enzyme has to be done in a proper way. You have to study a lot of things before designing the experiment. So once this cutting process is done, you have cut the plasmid vector, you have cut the foreign DNA with the same enzymes. So the sticky ends are generated. Next step will be to join them. And for joining, as we already discussed, you need to add the DNA ligase. Okay? And that process is called the ligation reaction. So the joining process is done. After this joining is done, that means you have one recombinant vector formation. Let's say the joining has been done successfully. So what you generate is a recombinant vector. Now what you have to do? You have to introduce the recombinant vector into a bacterial cell. That is, in this case, we normally, as I said, that it is because Escherichia coli or E. coli, it is a very fast growing bacteria. The generation time is 20 minutes. So it multiplies very fast, and it is very much used in laboratories for the cloning purposes. So you introduce that recombinant vector into bacterial cell, and that process is called transformation. So how do you do transformation? Normally the bacterial cells, you subject them to a heat shock. So first you prepare competent cells. You make the bacteria competent for uptake through treatment of calcium chloride, okay, which creates some sort of leakiness in the membrane. Okay, some pores. Although there are several hypotheses or several there are several propositions regarding the mechanism, but one of the mechanisms is they create some sort of leakiness. Once this is done, then you keep it overnight, and the next day you add the recombinant vector into the competent cells. Keep it on ice and then subject them to a heat shock for 90 seconds. Sorry, at 42 degrees centigrade, yes, for 90 seconds. But the temperature should be at 42 degrees centigrade. Fine. Immediately chill it on ice and then you plate it. Plating should be done in presence of antibiotic selection. That is, agar plates which have been already, where you have introduced the specific antibiotic which was present in the vector backbone. And you grow the cells. If the cells grow, that means definitely the vector has been introduced into the cell. But if the bacterial cell failed to grow in the antibiotic, that means it has not uptaken the foreign DNA. So that will give you an indication that the cells are positive. But don't stop there. Finally, what you do is you select the clones, right? And then you grow an overnight culture. And from the overnight culture, what you do is you re-isolate the DNA, right? You re-isolate the plasmid DNA and then you check it. That is, in this case, you have to check it through agarose gel electrophoresis. So you run the isolated DNA on an agarose gel and you can compare against what? You can compare it with a non-recombinant vector. So you run a non-recombinant plasmid vector and in parallel you run a recombinant vector. Because the recombinant vector has a gene of interest being introduced into it, so it will migrate at a lower rate because the molecular weight is high. So it will migrate slowly and the non-recombinant vector will migrate at a faster rate. So what will happen? The non-recombinant vector will show a lower band in the agarose gel and the recombinant vector will show it will show a band at a somewhat higher position as compared to the non-recombinant vector because there will be a shift or there will be a retardation in the migration rate. So in that way you can check. Another way of checking is that you can simply undergo a polymerase chain reaction again. You isolate the recombinant vector and you use the gene specific primers, the 5' primer and 3' primer, you use them to see if the gene of interest is being amplified from that recombinant vector. So if the gene is amplified, you will get a PCR band, right? And that means it will give you an indication that yes, the gene of interest is there in the plasmid vector. So you can just undergo a PCR reaction using the isolated recombinant vector molecule. You can run it again on an agarose gel and you can check the presence of that particular band. So this is another indication where you will get an idea regarding, yes, your entire work is successful. Your entire cloning process is successful. Of course, there are several complications in this cloning procedure also. Sometimes you need to clone more than one component. Like you need to clone 2 or 3 genes together. So in this case, it is a bit complicated issue. But I'm not talking on those issues because those are more advanced level knowledges. Okay. Maybe as you move into your postgraduate courses, you might have to learn those kind of issues like transgene stacking, gene stacking or gene pyramiding, which I'm not talking here. Let's keep it simple. So learners today, so if we sum up what we have learned today is basically we have learned about the very basics of the cloning procedure. We have learned about the components of the cloning process, the enzymes mostly, the restriction endonuclease, ligase and the alkaline phosphatases, and then what are the steps and also the vectors, mostly the plasmid vector, their criteria, their characteristic features, and finally what are the different steps of the cloning procedures, how do we use these components in the cloning procedure. So I hope you enjoyed this session. And you please go through your units, you please go through your blocks, and it's a very, very interesting topic which is very common. Molecular biology, biotechnology, it's a basic technique, and if there are any questions or you face any problem, please feel free to contact us or write to us or contact us. Thank you all and have a very, very pleasant learning. Thank you.